All right, put your cigarettes out. It's time to get back to talking about biology. Just for posterity, there are going to be some people who aren't in class in the room with us right now. We've just started the Tegrity recording now. We're probably about 20, 25 minutes into the lecture, right? But this part's going to be recorded, so I'm going to have to bring this back to the PG mode and stop doing all the raucous R-rated stuff that was going on for the first 25 minutes. So again, those of you who weren't here this morning, you're just going to have to wonder, what may have been transpiring. But I've been talking about eukaryotic cell division, prokaryotic cell division, and there's one thing that just blows me away more than anything else about prokaryotic cell division, and that is what a feat it is to replicate those bacterial chromosomes. E. coli's 4 million nucleotide long circular chromosome gets replicated in on the order of 30 minutes, sometimes as little as 25. That means more than 2,000 nucleotides per second have to be faithfully copied to be able to successfully make 2 E. coli where once you had 1. The process altogether is called binary fission. All right, but let's get to where the complexity is. I say eukaryote, you say complex. I'm not saying it, but that's okay. I feel like you're leaving me hanging. So, let's ratchet up the complexity here a little bit. The trick to replicating a prokaryote turns out to be replicating the chromosome, right? The trick to replicating a eukaryotic cell turns out to be replicating the eukaryotic cell's chromosomes. For E. coli, you get a pass in the sense that if you replicate the cell and one of the daughter cells doesn't get the chromosome, life goes on. And in some sense, you could even argue it may be a good thing, at least from the perspective of the daughter cell who got 2 copies, because it can replicate twice as fast, or much faster now, since it's got 2 copies of its chromosome, than it would have if it had to wait to replicate that chromosome. For a eukaryotic cell, like yours or mine, if you get here's the trick. We don't have just one circular chromosome. We don't have circular chromosomes to begin with. That's a little bit of a challenge. But you and I have 23 different pairs of chromosomes. Even the silly little fruit flies whose babies' heads you were ripping off in lab last week. That was my idea to have you guys do that, by the way, because I had such fond memories of doing it myself in my own youth. But even those silly little fruit flies whose babies' heads you were ripping off, did you feel guilty, a little pain? Did you realize that they have 4 pairs of chromosomes? If you were really good at that, you may have seen that they had 4 pairs of polytene chromosomes. Here's the thing, if you're replicating a chromosome and you got just 1, getting 1 to both sides, it's kind of a 50-50 proposition, right, when you're doing the daughter cells. If you blow it, it's not catastrophic. The daughter that doesn't get it dies. The daughter that got 2 has a leg up on the next round of replication. That doesn't work for you and I. We have 23 pairs of chromosomes. When our eukaryotic cells divide, if we get daughter cells that have 22 copies of, we're missing 1 chromosome on the left-hand daughter cell, and we've got the full set of 23 pairs plus 1 extra chromosome in the right-hand daughter cell. Both of those cells are messed up, right? The one on the left doesn't have a complete deck. You're building a building, you got the blueprints, and you're missing 1/20th of the instructions, right? Probably not going to be a structurally sound building. You're going to be really lucky if the 1/20th that didn't get copied and given to the builders turns out to only be for the trim of the color on the outside of the building, right? And even still, you're paying for a building, get the trim on. That cell's got a problem. The cell that got the 1 extra chromosome, because things didn't work out quite right with that cell division, right? The cell that got the extra chromosome, it's in trouble too. There's this problem that it's going to have that's related to gene dosage. The way we regulate gene expression to a large extent depends on how many copies of a gene we have. If you double the number of copies of a particular gene, you'll double the amount of proteins you make from those genes, and things get out of whack, right? There's a fine-tuned balance here. We're fine-tuned biochemical machines, and if you're making twice as much of one protein relative to all the rest, that usually causes problems too. We got issues as eukaryotic cells for cell division, and those issues are ultimately rooted in the fact that we don't have just one chromosome, not just one big chunk of genetic instructions. We've got, for you and I, on the order of dozens of chromosomes, and we have to make sure that there's a copy of each that gets delivered to both of the daughter cells. That's our big problem. All the rest for eukaryotic cell division is, again, it seems, at least compared to that problem, it's fairly trivial. Making enough ribosomes? Yeah, sure, not that big a deal, right? You're going to make on the order of 500,000 ribosomes inside of a cell, right? And if one cell gets, 249,999 and the other gets 250,001, come on, seriously, is that big a deal? Even if you only got like 100,000 in one cell and 350,000 or 400,000 in the other cell, not that big a deal. But those instructions are going to be a real problem for us. And so for eukaryotic cell division, very much like it was for prokaryotic cell division, the name of the game is replicating the genetic instructions, and now, more so than in prokaryotes, making sure that the daughter cells get perfect and exact copies of the complete set of those instructions. Okay? So for prokaryotes, it's replicating the chromosomes. For eukaryotes, it's not just replicating the chromosomes, it's getting them equally apportioned to the two daughter cells. And in a word, that problem gets solved by the process that we call mitosis. And mitosis is fancy talk for nuclear replication. You have one nucleus. At the end of mitosis, you have a pair of nuclei. Latin, plural, right? So we go from one nucleus to two nuclei when we are doing eukaryotic cell division. We accomplish that. If you wanted to think about this in chemical terms, you've got the substrates on the left, the products on the right. The arrow in the middle has the word mitosis written over the top of it. That's the name of the process by which you go from one nucleus to a pair of nuclei. So here's what chromosomes look like. Come on, you guys have seen pictures like this many times before. At some level, for eukaryotic chromosomes, what you got to do is you start off with these kind of chromosomes. You have to replicate them. During the replication process, they're held together, the copies. Right? So these are two sister chromatids. That's a chromatid. It's a chromosome as well. When you replicate it, you've got two of them. They're held together at this junction called the centromere. And then after mitosis, what happens is that on one nucleus, you'll have one copy of the chromosome, and in the other nucleus, you'll have two. Remember, that is a nucleus, the picture that they're showing you right here. So you start off with one nucleus, you end up with two. The process is called mitosis. This is a simplification because this is suggesting to you, if you took it literally, it's suggesting to you that you've got one chromosome in your cell. You've got one linear chromosome in your cells. You've got 23 pairs. What you see there on the screen has to happen 23 times inside of each of those nuclei in order for you to go from one nucleus to two nuclei. All right? So, in a word, we accomplish this with mitosis. Let's put things into perspective here. Most of our cells spend their time not doing replication. Right? So for a fairly typical cell, you can see they're showing you here that The mitotic phase, all of that cell division stuff, is a little tiny slice of the pie. Most of our cells, most of the time, are hanging out in a phase called G1. G is short for growth. Okay? And they're just hanging out. They're doing what they need to do. You know, your neurons don't divide that much. Okay? You get born with a certain number of neurons. Some of them fade away the more you drink, the more drugs you take, right? You can hurt those. They don't replicate that often. They don't replace their numbers that often. And so most of our nerve cells hang out for our entire lives simply in G1. Once in a while, and there are some of our cells, our skin cells need to replace themselves all the time. Some of our cells move from the G1 phase into the S phase, and the S is simply short for synthesis, and it's during that synthesis step that all of the chromosomes are being replicated. You can see for this particular cell, they're suggesting to you it takes a fair bit longer to replicate our prodigious amount of DNA than it does to actually do the business of cytokinesis and mitosis that splits this new cell, the big cell, into two baby cells. Okay? E. coli has a 4 million nucleotide long genome that it takes about a half hour to replicate. You and I use essentially the same machinery that E. coli does at a molecular level to replicate our chromosomes, but E. coli has a genome that's 4 million nucleotides long. Your genome, of which you should be very proud, is 3 billion nucleotides long. It's about 1,000 times, come on, between friends, it's effectively 1,000 times more DNA needs to be replicated to copy your genetic instructions than it takes to copy E. coli's genetic instructions. If it takes E. coli 30 minutes to get the job done, and you're using the same stone knives and bear claws that E. coli is using to replicate your DNA, what's 1,000 times 30? It's 500 hours. Oh man, that's seriously rate limiting. Now there are tricks that we have. Having multiple chromosomes lets us reduce that time because each chromosome can replicate independent of the others. But nonetheless, to replicate our DNA is not a trivial proposition. Certainly not from the perspective of time. There's neat stuff you can do with your big 3 billion nucleotide long genome, but it comes with a price, and that price is it takes a long time to copy all of those genetic instructions. So you can see in this picture they're trying to emphasize that to you here by suggesting to you that once you've decided one of your cells needs to replicate, you'd better be prepared to set aside a good chunk of time to get that DNA replication accomplished. Typically, at a minimum, a dozen or so hours, right? E. coli can get the job done in a half an hour. You're not that nimble, right? It takes you hours as opposed to half of an hour. Pardon our dust, right? As we improve the facilities. And then you'll see that for a replicating eukaryotic cell, after the DNA synthesis is complete, there's often another lag before you actually pull the trigger and do the splitting of the cell into two, another growth phase, G2. The length of time a cell spends in G1 can be very variable, can be very different from one cell type to another. Some cells spend their entire lives in G1 for an adult organism. The length of time spent in G2 can also be very variable. Some cells, instead of parking in G1, will park in G2 so that when they get the signal, they can, bam, split and make two copies of themselves really fast. Right? So it's a strategic type of decision, and it's different from one cell type to another. But all of this stuff, G1, S, and G2, all of that stuff together is what's called interphase. As you see where the priorities were for the folks who are making these diagrams are, right? Bulk of the time is spent in interphase, but the action is happening in that mitotic phase when both mitosis and cytokinesis occur. I'll tell you right now, cytokinesis is trivial, it's boring, it's a cleavage furrow that splits the big cell into two daughter cells. There's not much more to it than that. There's not a lot of concern about making sure you get mitochondria into both of those daughter cells, because for goodness sakes, you've got hundreds of mitochondria. What are the odds that all 100 or all 500 would be on one side of the cleavage furrow and none would get through the other? If you get just one over to the other side, that mitochondria can repopulate that new daughter cell, right? Cleavage furrows, cytokinesis, trivial. Nuclear replication, or mitosis, not a trivial proposition. All right, so this is all together called the cell cycle. Mitosis is where we're going to be focusing our attention. Check it out. You started with one nucleus. At the end of this, you've got two cells, each with their own nuclei. And in a nutshell, what goes on with mitosis is this. There's a number of steps that you go through. Let me step aside for a moment here, right? I know from past experience that a number of things are probably going through most of your heads. One of those is like, hey, I've seen this before, all right? Yeah, sure you have. This is fairly standard stuff. Another is that, man, I had to memorize this stuff before. I don't like memorizing this stuff. Guess what? I think you kind of need to memorize this stuff, all right? But here's another thing that I can tell you right out of the gate. There's no other logical way to accomplish this. So I hope that during the course of the next 10 minutes or so, I can talk with you about why this strategy, going through prophase and metaphase and anaphase and telophase, why going through those steps just makes good strategic sense. And I'll defy you to come up with some alternative that makes more sense, or even as much sense. This is the only way to get it done. And so I'm going to draw your attention to what it is that needs to be done. And once you see what needs to be done, about the only way you can do it is the way that nature has handed us an approach to getting it done. Okay? And one last thing, a fourth thing, as I'm ticking things off on my fingers here. The words that we're going to be using, prophase, metaphase, anaphase, telophase, the words that we're using are convenient for us as people to talk about. They don't necessarily have biological significance. So your textbook will go on at some length about the difference between early prophase and middle prophase and late prophase. From a cell perspective, it's all just part of a continuum, and there's no bright line that you cross when you go from early prophase to middle prophase, right? It's just part of a process. It's just convenient for us when we're talking about, at the beginning of prophase, this has to happen. Don't get hung up on the precise, specific differences between these different phases. Rather, think of it as a process. For mitosis, there is only one point in the whole process that is actually an instant, that is unambiguous, and that, there's stuff that happens before that instant and stuff that happens after that instant. That instant is metaphase, right? So metaphase metaphase is a word we use to describe a point in time, a very specific point in time, and you could talk about the stuff that happens before metaphase and the stuff that happens after metaphase. There is no early metaphase, there is no late metaphase, right? It's an instant in time, and everything before metaphase is building up to metaphase, and everything after metaphase is cleaning up the mess that you made to get to metaphase, okay? So there's the whole process. Ultimately, remember, what we're doing here is we're going from one nucleus to a pair of nuclei so that we can do cytokinesis and split that mother cell into two daughter cells. Okay? A lot of the rest of what I'm going to be doing with you now is zooming in at each of these different steps in the process to let you get a closer look at what's going on. And I don't even need to give you guys a closer look at what's going on because you guys did this all last week in lab. Remember? That's what you were doing. You were looking at root tips and seeing how those cells have catching cells in different stages of this mitotic process. So the pictures up above here show you how closely, or if you prefer, you could think how less closely or how poorly these cartoons are capturing what's really going on during these processes. The cartoons are simpler and easier for us to look at, so I'll focus on them. But you can see from these actual photographs of dividing cells that the cartoons are fairly good representations of what's going on. So, here's what's up. During G2, in interphase, the chromosomes would have been replicated. Okay? Eukaryotic organisms, unlike prokaryotic organisms, are what we call diploid organisms. We have 2 copies of all of our genetic instructions. Prokaryotes, in contrast, are haploid. That means they have 1 copy of their genetic instructions, so 1 copy of the chromosome. But you as a diploid, when you're hanging out in interphase, for the most part, certainly in G1 of interphase, you've got 2 copies. You have 2 copies of each of your chromosome 1s. You have 2 copies of each of your chromosome 2. And I'm not going to go all the way through all 22 plus the X and the Y chromosomes, right? You get 2 copies of all of your genetic instructions. You're a diploid organism. During the S phase, DNA synthesis, you make the transition, your cells make the transition from being diploid to tetraploid. You have 4 copies of all of your chromosomes, at least until mitosis gets completed. All right? And so in G2 of interphase, just to remember where we're talking about here, in G2 of interphase, that comes after S, the S phase, when the chromosomes get replicated. So this nucleus that you're seeing here has a whole bunch of stuff, squiggles in it, that is 4 copies of each of the chromosomes inside of that cell. If it's a human being, you have 23 different chromosomes, 2 copies of each, 2 copies of each of those 2 copies, it's tetraploid. You know, one other thing that happened during the S phase that I haven't mentioned up until now is that this other little nibbin kind of thing inside of eukaryotic cells also gets replicated. When our cells are hanging out, they usually have 1 centriole, but during the S phase, the centrioles also replicate. So, as we make the transition into the beginning of nuclear division, what do you suppose has to happen? Well, the name of the game, the objective, we got 4 copies of all the chromosomes. We want to end up, at the end of this whole mitotic process, having 2 identical nuclei with the exact same genetic instructions. Okay? The trick that we've got is you got that pesky nuclear envelope that encapsulates the whole thing. Okay? So that's a problem that we're going to have. If we want to end up having 2 nuclei, that membrane's going to get in the way. Right? It would be neat if the membrane wasn't there. Then we can just take the chromosomes and move them to different sides of the cell, and poof, we're done. Prokaryotes don't have that problem. They don't have a nuclear envelope. They just move the chromosomes to the other side. They let diffusion carry the chromosomes to the opposite sides of the cell, and everything's fine. But we got that pesky nuclear envelope. You know what would be swell? It would be really convenient if when we want to replicate a nucleus, it'd be so convenient if we could just get rid of that nuclear envelope for a little while, right? Is there some way that we can just, it's nice to have for the most part, but while I'm doing mitosis, can I just forget about it? Can I have it go away? That's exactly what we do, okay? And so the very beginning of mitosis, one of the first things that has to happen, and I'll say this to you in fancy talk, right, so you can impress your friends at the bars later today, if that's where you choose to impress your friends, the integrity of the nuclear envelope begins to be compromised. Right? And what that means is the nuclear envelope starts to fall apart. All right? That's going to be a real big first step in getting this process to work. The nuclear envelope has to start to fall apart. So in mitosis, at the beginning, we got a word for the beginning of mitosis. That word is prophase. At the beginning of mitosis in prophase, one of the first things that has to happen is the nuclear envelope needs to get kind of patchy. It starts to fall apart. Okay? There are two other things that need to happen at the beginning of mitosis in order to get this job done. Got to get rid of that nuclear envelope, so it needs to fall apart. You also need to do something to make those big honking chromosomes that you're so proud of more manageable, right? It's not an unfair analogy to think of inside the nucleus, your chromosomes that aren't, cells that aren't dividing, they're long, string-like, sort of spaghetti-like molecules. They're all tangled up relative to each other. They're literally tangled up relative to each other. If you're picturing like a big bowl of spaghetti, you're pretty close. It's just that the spaghetti needs to be able to make knots and stick to other pieces of spaghetti for the analogy to really work. And imagine if you reached in and you pulled out one of those pieces of spaghetti, it's going to be all tangled up and tied up with other pieces of spaghetti, because this is spaghetti that makes knots with other spaghettis, and it sticks to other spaghettis in a really nasty kind of sticky kind of way. What's going to happen if this sort of nasty bowl of spaghetti with the sticky knotted spaghetti in it, when you're pulling up one of those strands of spaghetti, that spaghetti's going to break, right? If you pull on it too hard, you're going to have one big piece of spaghetti that's knotted up with a bunch of other spaghetti and sticking to it. You're going to end up with two pieces of spaghetti, probably both of which are knotted up and tied to other pieces of spaghetti. You know what would be really convenient is if your chromosomes weren't so sticky and knotted up and spaghetti-like. So why don't we do this? Just to solve this problem, why don't we, instead of having long string-like, spaghetti-like chromosomes, just for a little while during mitosis, let's condense those chromosomes down into a little bit more manageable, smaller chunks, less likely to tangle up with other chromosomes, easier to manage, right? Let's just do this. But seem fair, reasonable? Why don't we condense our chromosomes? Instead of having to be long string-like molecules, nice, dense, compact chunks of DNA, as opposed to long, stringy pieces of DNA. Let's do that. Let's condense our chromosomes. That's how we get this problem solved. That's another thing that has to happen during prophase. The chromosomes begin to be individually distinct relative to each other. They're no longer tangled up. They condense. They're easier to manage. So we got to do three things to get this mitosis underway. We got to get rid of that pesky nuclear envelope. We got to condense our chromosomes. And we need to work out some way to make sure that we get segregation, that we get the right split of chromosomes, that we get one copy of chromosome 1 on the left side and one copy of chromosome 1 on the right side of this dividing cell, right? We got to get a good segregation. We need to separate those chromosomes. The way that that problem gets solved is ultimately because of what the centrioles are doing. They're going to set up what's called the mitotic spindle. And for the mitotic spindle to work, the centrioles need to move to the opposite side of the dividing cell. So three things have to happen. And only three things need to happen. At the beginning of mitosis, fancy talk, prophase, the chromosomes begin to condense, right? See how they're spindly and all tangled up over here? Now they're showing you that these chromosomes are looking a little bit more distinct. It's easier to pick out one chromosome here than it is over here. The chromosomes condense. The nuclear envelope starts to fall apart. That becomes clearer the further you get into metaphase. And the centrioles start to move. They were together. They start to move to opposite sides of the cell. And this mitotic spindle starts to form associated with the centrioles as they're moving to opposite sides of the cell. By the time that you've been doing this for a while, the longer you've been lessening the integrity of the nuclear envelope, the less nuclear envelope there will be. The longer you've been condensing your chromosomes, the more condensed the chromosomes will be. The longer you've been moving the centriole to opposite poles of the cell, the closer they'll be to the opposite sides of the cell. All right? So the beginning of prophase is the start of that process. The end of prophase is the end of that process. If you want, you can call that prometaphase. Metaphase is the next step. So prometaphase. In my world, prometaphase is the end of prophase. Okay? If you want, you can call it prometaphase. I'll just do the translation. Oh, you mean the end of prophase, right? Tomato, tomato, aluminium, aluminum, right? You talk the way you want to talk. And in my world, I'm just going to think of it as the end of prophase. I don't care, right? I care that you know the process. And the process starts with prophase. In prophase, the nuclear envelope starts to go away, the chromosomes condense, and the centrioles move to opposite sides of the cell. Okay? At the beginning, that's all less accomplished than it is at the end. But at the end, what do we got? Well, the chromosomes will have been condensed as much as they can condense. The nuclear envelope will go away as much as the nuclear envelope can go away. You know how much the nuclear envelope can go away? It's gone, all right? And the centrioles move to opposite sides of the cell. You know how much they can move to opposite sides of the cell? They can get to the opposite sides of the cell, all right? At the end of that process, the beginning of mitosis, you're done with prophase. And an instant in time occurs. This instant is called metaphase. What happens at metaphase is all that stuff got accomplished. The nuclear envelope is gone. The centrioles are on opposite sides of the cell, and the mitotic spindle associated with the centrioles is fully developed. And the chromosomes are as condensed as they're ever going to get. Okay? And at that instant, when all three of those things have happened, it also happens that the chromosomes are all lined up on a single plane called the metaphase plate. Okay? When all of this stuff has happened, that instant in time is called metaphase. And so the beginning of mitosis is over at this point. And we can start talking about the stuff that happens after metaphase. The stuff that happens after metaphase is pretty much what you would expect. We need to pull those chromosomes to the opposite sides of the cell. The centrioles with their mitotic spindle is going to help us do that. We need to start to reassemble a nuclear envelope around those chromosomes. And that also is going to be something that you'll see happens as we play the rest of this process out. And you get that done, and then all you got to do is make sure that you get a cleavage furrow that causes the mother cell to divide into two daughter cells, each with a nucleus, and you're done. Okay? So in metaphase, you got your metaphase plate, and you start to go into this thing that we call anaphase. The difference between anaphase and telophase, it's entirely semantic. It's just words. We only have one word. Well, I guess if you want to be fancy, you could say we have two words for what happens at the beginning leading up to metaphase, prophase and prometaphase. To me, prophase, or at best, early prophase and late prophase. I don't know why they came up with two words for this one. This is the equivalent to prophase and prometaphase, right? The way they split it up into two. But you could say anaphase is the beginning of the process and telophase is the end. So we're just sort of splitting it into two here. There's no bright line. There's nothing that defines the beginning of telophase being different than the end of anaphase. They all just sort of blend together. All right? But metaphase is clear. We know when metaphase has happened. The next thing that has to happen is you need to start pulling these chromosomes to their respective corners so that you can make two new nuclei. And to do that, a couple things have to happen, literally two. One is that the copies of the chromosomes, those sister chromatids that had been stuck together at the centromere, that centromere needs to break. You need to be able to pull the two sister chromatids apart because we got to get a copy into both of the daughter cells. So the centromere needs to break. The sister chromatids then can separate, and you need to physically pull them to the opposite sides of the cell. And that physical pulling happens because of the shortening of the mitotic spindle that's attached to the centriole. So those little ropes, those protein tubes that are connecting the centriole to the chromosomes at their centromeres begin to shorten, and that in turn pulls the chromosomes, the sister chromatids, to the opposite sides of the cell. Okay? So after metaphase, you got to begin the end of the process of mitosis, and that involves breaking the centromeres apart, pulling the sister chromatids apart, and it also involves the beginning of the reassembly of the nuclear envelope. Okay? So at the very beginning, it's hard to see. There's not that much nuclear envelope there because, come on, give them a break. They just started. But by the end of mitosis, in other words, during telophase, what's happened is that the sister chromatids have been pulled pretty much as far apart as they're going to get. The mitotic spindle is pretty much gone because there's nothing more that needs to be done for pulling. They've already been pulled to the opposite sides of the cell. The nuclear envelope now starts to become more visible, right? It's becoming more and more back to what it was before mitosis began. And the chromosomes no longer have to be compact. They start to spread out again into their spaghetti, sticky, knotted sort of ways. Right? So after metaphase, what do you got to do? Pull the chromosomes to the opposite sides of the cell. You do that by shortening the mitotic spindle and breaking the centromere. Along the way, you can start to let the chromosomes get less condensed. Along the way, you start building new nuclear envelopes around both of those sets of chromosomes. At the end, where you're at is the mitotic spindle is gone. The chromosomes are on opposite sides of the cell. They're encapsulated by a nuclear envelope, and the chromosomes are kind of back to their diffuse, spindly ways that they were before we started mitosis. What do we got? We started with one nucleus that was tetraploid. Now you've got two nuclei, both of which are diploid. That's it. I feel like I should just throw the mic down and walk out, right? There's nothing more to say. That's what's up with mitosis. I'll do that one of these days. It's going to happen. I'm afraid I'd hurt your ears or something if I hurt the microphone. So check it out. That's what's been going on. That's this whole process. There's nothing scary about it. There's nothing, high tech. I love reading the student evaluations from any class that I teach. So I'm going to put a plug in for that right now. At the end of the class, with me handy a piece of paper, you get to say things about me. I wait for those things to come back. It's like a Christmas morning for me. I pour through those. Whatever else I'm doing stops so I can read those evaluations. And, don't fill in just the bubbles for the numbers. That doesn't tell me much. I love it when you write stuff down and you tell me things. And one comment that really, it hurts a little bit. I see somewhat frequently, maybe I see it because I tell you guys that people say this, it's like, that Crane guy tries to make simple stuff complicated. I sure hope you don't think I've done that to mitosis because, it is simple stuff and it's not complicated. If you think it's complicated, it's just bad on me, right? Don't let my butchering of this simple process make you think it's tricky. Just look at it. It makes sense. Is there another way you could get this done? Could you do this without dissolving the nuclear envelope? Seriously. There's a problem. There's one solution. Get rid of it for a little while. Could you do it without pulling the chromosomes to opposite sides of the cell? Don't be counting on dumb luck or, chance to get that job done. You got to have some physical mechanism. It's the centriole with its mitotic spindle. Could you do it without condensing the chromosomes? I don't think so. So what do you do to solve that problem? Oh, we condense the chromosomes. It's simple stuff. There's a couple of simple fundamental problems. The strategies for solving those problems are not particularly tricky or complicated, right? None of this stuff should scare you. Only thing that might trip you up is, is prophase, what's prophase relative to anaphase? And the way that I would remember that, the way I do remember that, is prophase happens before metaphase, anaphase happens after metaphase, and telophase happens at the very end. And cytokinesis even more at the end than that. Here's cytokinesis. When you look at this picture, what do you see there, right? I don't know. I think the word cleavage is kind of apt, right? You can tell what people were thinking when they came up with that word. That's a cleavage furrow, right? You take it like a knife and you're slicing it down the middle, right? That's what I mean by cleavage, right? You're chopping it right down the middle, okay? And so there's this cleavage furrow. You know what's going on there? You got that big bag, right? The big bag that is the phospholipid bilayer. On the inside of that bag are some microtubules that go across like the whole equator, and those microtubules just get shorter, and they're pinching the bag from the middle, from the inside in the middle, into a constriction that ultimately pinches apart, and you got two bags where once you had one. That's cytokinesis. Cytoplasm kinesis, movement. It's not that the cytoplasm is moving so much, it's that the phospholipid bilayer is constricting in the middle, and you end up with two separate bags of cytoplasm where once you had one. All right. In a nutshell, there's some pretty pictures, not that different from the pretty pictures that you guys saw yourself with your own eyes in lab last week. Oh my gosh, I make you see all of them over again. I tell you what, if you really want to look at those things that are on PowerPoint, that's on the pilot page for the course, I could talk you through it, but I think I'd be insulting you after what I've just told you 2 or 3 times already. It's not that tricky. You start with prophase, you make it to metaphase, and then you undo that stuff, and you pull the chromosomes apart and build more nuclear envelopes. One last thing. We're kind of in the home stretch here. Let's talk about how this process gets controlled. For eukaryotic cells, control of cell division is a pretty big deal. For a prokaryotic cell, control of cell division, not that big a deal. For a prokaryotic cell, pretty much the name of the game is divide as much as you can get away with dividing. Make as many copies of yourself as you can. That's what prokaryotic cells are in the business of doing. You and I have to be more strategic with the way that we do our cell division. Now, there are some eukaryotes that can adopt that prokaryotic strategy. Yeast, a single-celled eukaryotic organism. It can play the game the same way that E. coli does. But for you and I, there are certain cells that we want to have dividing that works well for us if they're dividing. And there are certain cells that if they start dividing, or if they start dividing out of sync with other cells nearby them, cause a problem. And we got a fancy word for that problem. It's called cancer. And so uncontrolled cell division in an organism like you and I, there's no hyperbole, right? That is the definition of cancer. Okay? When some of your cells, some of your cells start to not follow the rules and divide when they shouldn't be dividing, that's a big deal. That's cancer. And so, how do we make sure? What do we do as a fancy, highfalutin, complicated eukaryotic organism to prevent our cells from dividing when we don't want them to divide? Again, there are some of our cells that we want to be dividing pretty much all the time. There are cells in our bone marrow that we need to have making red blood cells pretty much full tilt, nonstop all the time. Doesn't matter too much what else is going on, just keep cranking out those red blood cells. The cells in our skin, the epidermis, they need to keep, we're sloughing off skin cells all the time. Every fingerprint has on the order of 100 cells associated with it. Did you know that? Right? You know how many cells we need to generate a DNA profile to connect you to a crime scene? 10 gets the job done really well, right? 100, that's an abundance, right? So, we're shedding cells left and right all the time off of our skin. We need to replace those. Our skin cells need to be dividing. you know, your liver cells, not that much. We don't need that many more liver. Your liver is probably doing just fine, right? And you don't need to replace those liver cells that often. You're probably good not having your liver cells divide. If they start to divide just because of the joy that they get from dividing, you may be getting a liver tumor. And what's probably, hey, it's not funny, liver tumors are serious business. But what's happened to those cells that our cells that get the signal to divide is there are 3 opportunities that our cells take advantage of to put the brakes on things. They're called checkpoints. And there needs to be an affirmative signal that the cell gets before it can proceed through the checkpoint. They come to a full stop in this cell cycle when they get to these checkpoints, and something special has to happen to get you past that checkpoint. Right? So there are 3 checkpoints. You can see where they are during the course of the cycle. There's the G1 checkpoint. What part of the cell cycle do you suppose the G1 checkpoint happens? Oh, how about in G1, right? As opposed to the S phase, right? That makes sense. The G1 checkpoint's in G1. Hey, check it out. There's also a G2 checkpoint, and very conveniently, it's within the G2 part of the cell cycle. It happens to be right at the very end. This one, to be able to start mitosis. And what happens at the start of mitosis? Oh, yeah. The nuclear envelope starts to fall apart. The chromosomes begin to condense, and the centrioles move to the opposite side of the cell. Before any of those 3 things happen, the cells need to get a thumbs up from at least 2 different proteins that, you know, it's like the nuclear missile silo sorts of things, right? Insert your key now, you know, turn the key to deploy, you know. They have to be done in concert by 2 separate things for like a fail-safe sort, so you don't have like a rogue, you know, nuclear weapons officer that decides, yeah, let's just launch my missile today, right? You got to have 2 guys. Yeah, or accidentally press the button. You don't want this stuff to happen just by chance. You want to have that double check. And so, before you begin mitosis, you got to get these 2 thumbs up. In the middle of mitosis, so just after metaphase, there's another opportunity where you have to have 2 thumbs up to be able to proceed through the process. And then most of the cells are hanging out in the G1 checkpoint because that's just where they are. They're just cells growing, doing their thing, metabolizing, squirting out insulin, whatever their job happens to be, they're hunkering down and they're doing their day-to-day work. Okay? So we got these 3 checkpoints. If a cell develops a mutation that causes it to start making the protein that gets you past the checkpoint all the time, not when it is that that protein needs to be made, you're a step closer toward getting cancer, right? And here's the thing. It turns out to convert a normal cell to a cancerous cell, interestingly, a minimum of 2 mutations have to occur. Okay, you can't do it. There's no single gene that you can knock out. There's no single protein that you can squirt into a cell that lets it get past all these checkpoints. There have to be at least 2 to convert a normal cell to a cancerous cell. But if you choose the right 2, you can ultimately set up a situation where these checkpoints will no longer apply. Here's some of the kinds of proteins that might get you to that point, right? There's this protein called cyclin. There's another one called mitosis promoting factor. If you get a mutation in both of those genes, well, one of your checkpoints isn't going to work anymore, okay? And you can see as you're going through the cell cycle how it is that the concentration of these proteins in the cells are changing in a way that you would kind of expect they would have to change in order to get past at least that mitotic checkpoint. So there was that one checkpoint at the in the middle of mitosis. Well, to get past this one, these are the 2 proteins that you need to be able to blow past that particular checkpoint and proceed along. And you can see after cell division, the concentrations go down, but when you're back to that point, if the cells are going to make it through division, both of those things have to go up again. Pretty picture of how it is those proteins are interacting with each other, and then coming to the different checkpoints. And again, I'll end on a happy thought that if that system breaks down, you get cancer. All right? Tell you what, because I had to launch today's PowerPoint off of my email, as opposed to Lauren doing it the way that she does it, I don't have a question at the end here for the last slide, but I'll tell you this. The question that I had typed in at the end was the very first question in the list of possible questions at the end of this lecture, right? So there's that, there's where I say words, and then I had something about questions to consider. The one that I had typed in was the first one that was on that list. It was something about binary fission, and you'll recognize it when you see it, all right? If I think of it, we'll start class on Thursday with that slide, all right? But other than that, let's call it a day. See you guys on Thursday. We'll talk about sexual cell division.
So recently